Good day, Junior Techies. Welcome to this video where we're going to be looking at question 7 of the worksheet dealing with DNA, nucleic acids. We've worked through a few questions here already, and hopefully you've joined me for those in some of the previous videos. I also hope that you have downloaded the worksheet and the worksheet memorandum just to help you prepare for your final exams. So here we're looking at question 7, and question 7 is a great question for us to get a better understanding in a few things like exam technique, question analysis, and also a little bit of the theory that goes along with it. So I'm glad you've joined me. My name is Mr. P, and let's dive into question 7. Right, so here we're going to look at the question to get some context. The sequence of amino acids in a protein molecule is coded for by DNA and RNA. Now, the DNA we know is found in the nucleus, and the RNA transcribed in the nucleus has to move to the ribosome in order for us to form the protein molecule. That is the sequence of amino acids referred to there. Okay, right. Now, in the table, we see mRNA codons and their corresponding amino acids, and I can only see on this page here question 7.1 and 7.2, each one mark, so I'm not thinking too much of those two, but this is a much larger question. Okay, we're going to find a few more questions coming up a little bit later, but let's get the context right first. Okay, so if we look at this again, and we're looking at the mRNA codons and their corresponding amino acids, how do we get to this point? Well, it all starts with the DNA triplet code, right? The DNA triplet code is going to form a very specific mRNA codon once transcription has taken place. My mRNA strand with my sequence of mRNA codons is going to move to the ribosome, and I'm going to have tRNA anticodons bringing specific amino acids that complement the sequence on my mRNA strand, and this is going to result in amino acids being in that specific sequence when my protein has been formed through protein synthesis. Okay, now let's have a look at question 7.1. According to the table, how many codons code for phenylalanine? Now, I know that's a difficult word to pronounce, so read it a few times, pronounce it a few times. Phenylalanine. Okay, there's my amino acid. Now, if we're looking for how many codons, we've got to go look at the amino acids first. All right? And here we're going to see that we have our amino acid in 2 positions in this table, and each is coded for by 2 different mRNA codons. So here we have 2. What is the anticodon for glutamic acid? So here I've identified it already, glutamic acid, the last amino acid in our list. And I have the mRNA codon as GAG, but it's not asking me for the mRNA codon. It's asking me what is the anticodon, which means I'm talking about the tRNA. All right, so what is the complementary tRNA codon going to look like? There's my anticodon, CUC, and that is the answer to question 7.2. So there is a little trick to answering some of these questions. You have to read your question very carefully, and sometimes the terminology does confuse us. We have mRNA codons, we've got tRNA anticodons, we've got DNA triplet codes, and we've got to know where those amino acids fit in, because my tRNA anticodon is going to carry a very specific amino acid. And it's going to place it in a very specific place on that mRNA strand when we get to translation at the ribosome. Right, so let's have a look at the further questions here. So here we've got a little bit more context as we move through the questions here. So a section of mRNA has the following base sequence and is read from left to right. So here we've got GAU, CUC, GAC, AGC, AUG, and ACC. This is my section of mRNA from left to right. Now let's look at question 7.3 when I relate it back to the context that I've just been given. Give the DNA base sequence for the last codon in this section of mRNA. So now we're working backwards. If this is an mRNA, we need to know is where did this mRNA come from if we refer back to the DNA. So here, where they're referring to the last codon, ACC, and if I take it back to my DNA, ACC would have resulted from TGG on my DNA. So there's the base sequence for the last codon on this mRNA strand. Now, next question, 7.4. The first amino acid coded for by this section of mRNA. So now we're looking at that one. And if we take it back to our table, which is also giving us a lot of context, it says my first amino acid. So now I've got an mRNA codon, so I can go with GAU. I go to my table, I look for GAU, and I get to aspartate. Right, so that is the amino acid coded for by the first mRNA codon in this sequence. Okay, brilliant, not too tricky yet. Now, 7.5. Now I get to a different context. A mutation occurred which resulted in the forming, or resulted in the following, base sequence of mRNA strand. So now, let's have a look at how this compares to the sequence we have already worked with. So we've got the first mRNA codon is the same, the second mRNA codon, CUC, is the same, third GAC is the same, the fourth we've got as part of the first sequence AGC, and now after the mutation has taken place, I have AGU as my fourth mRNA codon. Let's look at the fifth, AUG, AUG, ACC, ACC. All right, so that's the only mutation that I see here. So my mutation occurred here. Okay, great. Now let's look at the questions. Describe the mutation. So in other words, what I've just identified, I need to now put into words. What has happened? So my fourth codon is where the mutation took place. And what happened? C was replaced by U, and that is the mutation that has taken place. So there are my 2 marks. B, explain the effect that the mutation will have on the resulting protein. 2 marks. Now, we are not going to jump to our default answer here, because in many questions, we know that, right, so because of the change in base sequence, we change the amino acids, therefore we are changing the resultant protein. But we need to make sure that that is exactly what's taking place. So looking at our context, AGC and AGU code for the same amino acid. So let's have a look now. How is this impacted on the final protein, or the protein that's being formed? Well, the protein is actually staying exactly the same, because the amino acid carried by each of those tRNA complementary anticodons, so complementary to AGC and AGU, it's the same amino acid. So how do we answer this question? Well, both AGC and AGU code for serine. So this will not impact on the resulting protein at all. The resulting protein will be the same. And there's my 2 marks. Now, question 7.6. Let's have a look at this one quickly. Right. Name and describe the process. Name and describe. There are 2 parts to this. I have to name it, and then I describe it. The process that takes place in the nucleus, now there's only one in protein synthesis that takes place in the nucleus, and this is transcription, that results in the formation of an mRNA molecule. So here we're definitely referring to transcription. So I can name it, and then I need to discuss the process. So transcription first is so important, because it asks us to name it. There's my compulsory mark. Now we are describing the process of transcription. So very similar to a previous question we've had in this section as well. But let's have a look at how many points we can get here, how many marks. So the double helix DNA molecule has to unwind. Weak hydrogen bonds do have to break for the DNA to unzip itself. Okay? And I'm forming two separate strands once it unzips itself. In the forming of an mRNA molecule, only one strand is used as a template. And that is so important. One strand is used as a template. Now this is my question to you, when are two strands used? Two strands are used in DNA replication, and that's not DNA replication that we're talking about now. It is mRNA formation, which is transcription. So one strand is used as a template. And in forming this mRNA strand, I'm using RNA nucleotides. Super important here to remember that it is RNA nucleotides. We're not referring to DNA nucleotides. Also, simply writing nucleotides might not get you that mark. So we need to make sure we're specific in the nucleotide that is being used here to form the mRNA strand. Also important to say that mRNA is complementary. So in other words, the sequence of DNA and the sequence of mRNA are complementary to one another. So the pairings are A to U, C to G, T to A, which is not written there, okay, and G to C. And this process is it's controlled by enzymes. Now, the specific action of each of these enzymes, not so important for you to name the enzyme and its action, because if we make a small mistake there, we might not get full credit for what we write. Okay, so be super careful there. If you're not sure of the exact role of the enzyme in this process, rather just write enzymes generally. Okay, that the process is controlled by enzymes and we're not losing marks because we're actually only asked to describe the process of transcription. We're not asked for the functions of each of the enzymes. All right, thanks Junior Tikkies. Thanks for joining me for this question, question 7. Again, a very nice question in applying our knowledge. Doing some exam technique here, looking at the question, exactly what the question is asking us for, making sure that we're writing and addressing each part of the question is important for us to get full marks at the end of the day. So keep practicing, keep working hard, and if you haven't yet, get that theory booklet, have a look at the worksheet, download that memorandum, work through those questions to prepare yourself, and I do hope that you do very well. All right, thanks Junior Tikkies. I hope to see you again.